Good evening from Colorado, and welcome to the inaugural webinar of the COMMeND Network for the second year. For our webinar today, we will be discussing non-typeable Haemophilus influenzae, which is the most common bacterial pathogen for otitis media around the world. Our first speaker will be Kevin Mason. Kevin is our current ISOM president, the president of the International Society for Otitis Media. And his lab has identified mechanisms of innate immune resistance and nutrient uptake that are essential for NTHi pathogenesis. His lab also seeks to understand how host nutritional immunity influences NTHi biofilm architecture, invasion of host epithelial cells, and modulation of inflammatory responses that contribute to NTHi persistence and disease severity. So Kevin is a faculty member of the Ohio State University. Please go ahead, Kevin.
Great. Thank you, Reggie. And thank you, Landon, from behind the scenes of putting all this together. It's a pleasure and an honor. And Reggie, congratulations on the great news of receiving year two for the grant itself. And so it's really fitting. I think that this Tanya had mentioned about the meeting and how great the COMMeND seminar was, or the symposium was, that session. And I totally echo those comments. And it's really great to see that the trainees, the level of the quality of the trainees and their presentations is so exciting. So I hope to do it some justice, some work that we've been working on. And what I thought I'd do this evening, morning, afternoon, wherever you are in the world, is take you through a bit of some of the approaches that we're working currently in our laboratory. Some of this is dated information pertaining some findings that I found during my postdoctoral years, but it brings it to some current unpublished work that we're currently working on, all really leaning towards a link towards development for some advanced therapeutics, really targeting some of these systems. So as Reggie had mentioned in the introduction, I really study my favorite organism, and that's non-typeable Haemophilus influenzae, and really from the standpoint of mechanisms of disease, particularly related to nutrient acquisition. We kind of stumbled into this area in my postdoctoral years with Dr. Bakelitz. Some of the early work, we did some work in the chinchilla model and really identified some of the early, some of the promoters of genes that were turned on during acute infection. And really that drove us down a pathway of antimicrobial peptide resistance that's conferred upon Haemophilus, but also now dovetails into a nutrient and iron uptake mechanism. So I'm going to take you through a few different stories here and try to wrap that up with a therapeutic angle. So let me click. We're not going. There we go. There we go. So I don't need to belabor this point, but obviously non-typeable, and Reggie mentioned this is really the predominant pathogen in otitis media now, major causative agent, but it's really not isolated to the ears itself, or the middle ear, but it's also a pathogen of both the upper and lower respiratory tracts. So the findings that we do have an animal model of otitis media. However, we're thinking about other animal models or other models type systems, both in vitro and in vivo, to study lung infection because non-typeable strains are also very significant in exacerbations of COPD in patients. So the mechanisms we describe in the ear, we also are looking at in some in vitro type model systems to see if they're relevant on lung cells as well. And so moving that towards more of just the upper airway. Haemophilus is pretty unique. It is a heme oxytroph, meaning that it must acquire iron through, or heme molecule through transport from its environment. And so that's pretty critical. Haemophilus cannot synthesize that molecule. So it's really dependent upon mechanisms to acquire that. And I'll highlight that point in a few slides. But non-typeable Haemophilus influenza strains are very important, not only during acute otitis media, but also prevalent in recurrent otitis media. The incidence rates there about 11%. Chronic otitis media is a big player. And this idea of persistence meaning longevity of infection. And so once Haemophilus gets to the upper or lower airways, it can maintain infections for a period of time, usually until the host can clear that or tries to clear that. But Haemophilus adapts quite nicely to these environments, and you're going to hear from our second and third speakers some nice work related to more aspects of that. We'll focus this part of the talk primarily on how Haemophilus will adapt to those environments. So the clinical problem is obvious. We just came off the otitis media meeting in Philadelphia. But this is the problem, right? And so the kids, some kids are very susceptible to otitis media, middle ear infection. Some kids acquire maybe 1 or 2 episodes. But the problem here is the sequelae of that disease, and ultimately still being the #1 driver for hearing impairment downstream, and then all the consequence of that. Here you see a middle ear that is really full, inflamed of middle ear fluid that ultimately leads to perforation of the tympanic membrane. One way to alleviate that, and still the #1 reason that kids are not only going to the emergency rooms with treatment of antibiotics, but with treatment with tympanostomy tube placements, and that's really to alleviate the symptoms themselves. If this is unalleviated, those tympanic membranes can rupture, and you see there in the far right image that kids can, this fluid will run, it's pretty much infectious fluid, can run from ear to ear. This is really a problem in some areas of the world where treatments aren't as readily available. So really thinking about how bacteria adapts to these environments really is a focus of my laboratory. And as I mentioned on the outset, this idea of persistence, really from the idea, really driven by multiple influences. It can be antibiotic therapy. In this case, might be familiar with development of persister type cells that would be have some sort of response to antibiotic therapy. However, oxidative stress, nutrient limitation, and tryptophan signaling, indole signaling, big players in really driving this idea of persistence of these populations in the ear. So we're really interested in this idea then of this how pathogens will evolve these systems. In this case, highlight the word elegant because you're going to see that we didn't know our work was going to go down this path, but bacteria very smartly utilizing a single system for multiple functions becomes very intriguing. Very difficult to study, however, very intriguing in the outcomes. And so we really think about that a lot, but then from the standpoint of how do we exploit those, and how does a bacteria exploit those systems to take advantage of those environments in vivo, and then ultimately survive and persist in those environments itself. So a few years ago now, during my postdoctoral work, we really stumbled upon this ABC transport system. And here I've depicted that here on the bottom of this figure. And I think you'll see that. I hope you can see that. Do you see that? Yeah, good. It's really I depict 7 proteins here, but it's an intermembrane transport system. It's an operon that's encoded in the Haemophilus genome. And up until this point, it was really unknown why this transporter would be present on the inner membrane. We did know that in other organisms that mutations in proteins of this transporter did confer sensitivity to host antimicrobial peptides. Those are depicted here in this cartoon model. These are depicted as human beta defensin 3 molecules. These are small cationic peptides that are produced by the host, really in response to bacteria that encounter these environments. Their job primarily is they're attracted to the membrane, they poke holes, and ultimately lyse those bacteria. Well, bacteria are very smart, and Haemophilus has acquired a means to deal with these antimicrobial peptides. So if I just have you picture here on the outer membrane surface, you'll see there's initial retraction mechanisms that occur where repulsion mechanisms, I'm sorry, where Haemophilus can change its surface charge and then repulse the cationic peptides and really prevent them from binding that surface. And that's a pretty good defense mechanism. However, during infection, these peptides build up. They will poke holes, essentially the hydrophobic components of these peptides will poke holes in this membrane and then gain access to the periplasmic space itself. Once here, if they do encounter the cytoplasmic membrane, they also will incorporate into that membrane, ultimately cause a disruption of that membrane potential, and then it'll cause lysis of the bacteria. However, here sits this SAP transporter system, and this was named for sensitivity to antimicrobial peptides because others, and we have shown that this periplasmic binding protein will in fact bind these cationic peptides, and then this serves to deliver them and transport them through this transporter into the bacterial cell, ultimately for peptide degradation. So in a series of work a few years ago, we were able to demonstrate that Haemophilus can actually grab hold of these peptides, transport them in, degrade them, and ultimately use them as a nutrient source, a peptide recycling source. So very smart, very efficient. The system is upregulated upon exposure to antimicrobial peptides. Well, not long after that, we published work that, because I have a slide here on some phenotypes that were observed. We made some mutations in this strain, and then it conferred some changes in phenotypic behavior of Haemophilus and how it interacts with host epithelial cells. Well, we discovered that SAP is actually part of a family of ABC transporter systems that will bind multiple types of substrates. And so in this case, it's very diverse, not only binding small peptides, but in other family members, the oligopeptide binding protein has been shown to bind the molecule heme iron. So at the start of this talk, I had mentioned that Haemophilus is a heme oxytrope and needs to acquire heme iron. And so because of this, this became a very attractive system that perhaps this was the mechanism that Haemophilus could acquire heme iron across that inner membrane. And so, in fact, we did some experiments with that, and that, in fact, is true. And so this diagram cartoon becomes a little more complicated. So in this case, now you see SAP-A not only interacting with antimicrobial peptides, but also interacting with the heme iron molecule. We know from some works that antimicrobial peptides can actually displace the heme iron molecule. And we've since then, through work with a collaborator, Heather Pinkett at Northwestern, we've crystallized the SAP-A protein and have shown that it not only binds the antimicrobial peptides, but we are now currently mapping those binding sites and those residues. But it became very important for us to think about this in terms of, if this is in fact true, how does Haemophilus confer these resistance to these peptides? And if that is true and we can define the binding site, can we ultimately design some peptide therapeutics that really target that binding site? And if we can do that, then that would also ultimately cripple Haemophilus and its ability to survive. Really this idea of an advanced peptide mimetic, if you will, which would substitute in place of the antimicrobial peptides. Well, we tested a panel of antimicrobial peptides, and we noticed, so what this is a survival curve. Here's the concentration. It's increasing from lower to higher of either the cathelicidin LL37, HBD3 defensin, HBD2 defensin, and then an alpha defensin human nucleotide peptide 1. And so what we were really looking for, we generated a SAP-A mutant, compared that to the parent strain, and you were looking to see if there's any change in sensitivity to killing of these peptides that was dependent upon SAP-A. And we did see this with the cathelicidin molecule, the HBD3 molecule, not so much with HBD2. There's not significant relevance here in these higher concentrations. And then human neutrophil peptide was there was no difference between parent and SAP-A, the SAP-A mutant, which suggested that there's a hierarchy of dependence upon which type of antimicrobial peptide and the resistance that is conferred. So that becomes very important as we start to think about this binding site. So this really brings us to our current work, and we published this last year in collaboration with Heather Pinkett from Northwestern. We have crystallized, as I mentioned, the SAP-A binding protein. And here we show all the residues that are predicted to interact with the human beta defensin 3. And then we're going through the different types of peptides to see if they're sharing binding sites and whether they overlap. And you can see there from panel D that we can actually take a amino acid approach. We do basically an alanine scanning of site-directed mutagenesis, and we can see if we replace one amino acid, does in fact this affect binding of HDB3 to the molecule. And some residues are important in that and some do not. What's important is that now we have this panel of candidates, and on the right you can see two inhibitors, and this is some encouraging results from maybe a couple of weeks ago, where now we have some inhibitors that we can block the binding site, thereby preventing binding of the antimicrobial peptide. And if you remember back to that cartoon model, if SAP-A cannot bind the antimicrobial peptide and then transport it for degradation, they will accumulate in the inner membrane and cause lysis of the bacteria. So in essence, this would be a therapeutic designed towards this peptide, toward this protein, in essence, to decrease or affect persistence of Haemophilus at those sites. So that's ongoing work, and hopefully over the next year or so, we'll really define that in these peptide inhibitors and show the effectiveness of that treatment. I'll shift gears just a tad here. Let me keep tabs on time. I had mentioned that when we made a SAP-A mutant, essentially we conferred phenotypic changes on Haemophilus that we really hadn't seen before. And here you'll see on the left in biofilm formation that a SAP-A mutant essentially looked different when we incubated it on epithelial cell surface. And that would be here with the SAP-A mutant. This would be the parent strain. Whereas the parent strain forms a nice biofilm on the surface, the SAP-A mutant really caused some disruption of this epithelial cell surface. We looked at this by transmission electron microscopy, and what we could see here essentially on the right is that the SAP-A mutant was more prone to invasion of those epithelial cells, and these are all bacteria that get inside the epithelial cells, multiply, and essentially will form these intracellular bacterial communities. This was a SAP, remember this is a SAP-A mutant itself. The parent did not do this. The parent kind of sat on the cell surface. Some of those did get inside vacuoles, but they were quickly lysed inside those vacuoles, ultimately being a dead end for any intracellular survival for the parent strain. So this really suggested to us that a disruption of this system could in fact change the behavior of bacteria, and possibly how it traffics throughout the epithelial cell itself. So now, if that is true, one of the next experiment was to really take the wild type strain and then expose it to a period of lack of heme iron, so essentially an iron starvation event. So that's what we did in these biofilm formations. What you see is either in green or red here. These are towers of biofilm, and then that's formed on a mat of a biofilm itself. These towers are both from a population of Haemophilus that had previously been iron starved. All we've done here is change the reporter, so from green to red fluorescent protein, really to show that the reporter itself wasn't causing this phenotypic change, but it really made an architectural change in how biofilms grow. So if you heme iron starve, not only do they appear to be invasive, but they're also changing architecture of Haemophilus itself. And so we quantitated that here in this idea of persistence. Invasion, we looked a bit more into this. In this case, we just use a GFP version of our bacteria, and then we could demonstrate that Haemophilus, if it was previously iron starved, will in fact infect, get inside epithelial cells and form these intracellular bacterial communities. And you can see in panel D here, the quantitative analysis that we had, I don't have time to get into it, but we had isolated a persistent isolate of Haemophilus, and that forms a lot of these IBCs really early during infection, and they're maintained over a 4-week period. Haemophilus, the wild type strain, makes some IBCs very early, and then you can see it increases significantly out by 28 days. So not only is Haemophilus getting inside these epithelial cells, but they're growing and propagating within them as well. So really that was an interesting phenotype, and we had thought about some mechanisms of perhaps why that may be occurring, and then how to study that. Well, about at the same time, we did an animal infection experiment. Once we noticed that these IBCs were formed in vivo in our chinchilla model, we asked the question, how long will those IBCs persist? And so in this experiment here, we infected chinchillas in the middle ear, and then over a period of time, we just monitored them over a period of weeks. You can see here on the left at week 1, those are the counts in the red, these being the IBC counts in those middle ears. The gray step down is an indication of the ears that had effusions in those middle ears. So this decreases over time, and we know this. Ultimately, by about 30 days, the fluid the host will clear the fluid, the inflammation will subside in those middle ears. But our question was, how long can we detect IBCs? We went out 7 weeks and we still could detect the presence of these intracellular bacterial communities into this ear. And the point being here is this was long after clinical resolution of disease. And so this is an active area in my laboratory now. We're really studying this population. Why are these bacteria here? And if they are here, can they be a source of recurrence of otitis media? And if so, what events cause those to recur or come back out? So we have a panel of studies ongoing now, whether viral exposure, smoke and e-cigarette exposure, or a secondary bacterial exposure will cause those bacteria to come back out and reinstitute a secondary infection in that middle ear. So those are ongoing studies. We have a grant out now, so hoping for some funding there so we can continue those studies. This is a bit complicated slide, but I just really want you to focus on the upper left here. This is the mechanism that we now believe leads to formation of these intracellular bacterial communities. Haemophilus can get in through either clathrin type mechanisms, caveolae or lipid type mediated endocytosis. These we believe are dead end. They get into these vacuoles, they're targeted to lysosomes, and the bacteria will be removed from the epithelial cell. However, if they do come in through this macropinocytosis mechanism, we do know they evade lysosomal fusion. In this case, will result in IBC formation. The reason why I have this slide here is this bottom left. We have identified an inhibitor that will block macropinocytosis. If this is true, then we should be able to prevent the formation of IBCs, and that's in fact what we demonstrate here. So on the left, this is an intracellular bacterial community that has developed in the middle ear of a chinchilla. If we treat with EIPA, this inhibitor, we no longer see the formation of these intracellular bacterial communities. And this is just quantitated here in the bottom. This is significant because if we can then, through therapeutic treatment, prevent the formation of these intracellular bacterial communities, then the bacteria are going to be more susceptible to either antibiotic treatment and or just the host innate immune system, which will ultimately clear the bacteria. So likely a combinatorial type treatment, but nevertheless one that targets that mechanism of entry to ultimately drive towards some advanced therapeutic. Okay. What are we looking at, Reggie? About 10 more minutes? We started a little late. I want to make sure I get through this part.
You can go ahead.
Good. Okay, so this idea of nutritional immunity, this really then shifts our focus a bit more to this understanding, because we identified a transporter that can do multiple things. It can deal with resistance to antimicrobial peptides, so really providing mechanisms of resistance to innate immune component, but at the same time be a transporter for bringing in heme, which really has to be brought in from the environment in itself. It really sets up this idea of nutritional immunity and how the host serves to sequester heme and or iron in multiple compounds, really to starve bacteria, even beyond Haemophilus. Many bacteria are susceptible to this idea of nutritional immunity through sequestering of essential nutrients, ultimately leading to the death of the bacteria. However, Haemophilus is quite smart, as I told you. This is obviously a very complicated slide, but I just want to focus you to the outer membrane here itself. So this is the inner membrane, this is the outer membrane, and this is what we currently believe is going on with Haemophilus as it needs to acquire iron sources. Obviously, Haemophilus has heme binding proteins, HXUA, that can be soluble also in the membrane itself. HXUC, these are heme binding proteins. They will bind heme and transport them into the periplasmic space. Haemophilus also has transferrin binding proteins, so they can capture iron that's complexed in transferrin and then acquire iron this way and then transport through iron transporters. A huge source of iron, obviously, is hemoglobin haptoglobin. So hemoglobin is a molecule in our red blood cells. It contains 4 heme molecules bound in complex, but hemoglobin does not exist alone. It binds haptoglobin in the human body. So this complex of hemoglobin haptoglobin must be recognized by Haemophilus, and it has 3 known hemoglobin haptoglobin binding proteins, HTB-B, C, and D. So essentially, hemoglobin haptoglobin will bind. heme is stripped and the heme is transported, and then we believe this gets in through the sap transporter into the space itself. Well, the reason why this is important because we think about these in terms of how do we leverage a bacterium's need for these nutrients as an approach to help mitigate the disease process itself. And is that a potential? I had alluded to some of these experiments earlier regarding restricting heme iron, restricting Haemophilus of heme iron itself, or as a compared to just having heme iron all the way around. So we table that continuously exposed. So in this case, the question became, could we essentially starve Haemophilus for a long period of time? And then we were really interested to see if this promoted any changes genetically that would affect persistence of Haemophilus itself. Well, you can see here in this graph, if Haemophilus is heme iron exposed, and that's the light gray bars in the back, it essentially will survive continuously, right? This is to the point where I had a graduate student working on this, and she ran out of culture on day 39, so we couldn't look beyond that. However, we sequenced at all these different points, and we did notice there was a single mutation in the strain that occurred about day 33, and that was in a cyclic AMP phosphodiesterase, but that essentially then conferred a persistent type phenotype upon our Haemophilus strain itself. This resulted in multiple changes in Haemophilus metabolically. So we saw an essentially shift. This persistent isolated shifted into, it would be the light gray bars here. If you're looking at increases in amino acid production, at all cases, we notice there's an increase in amino acid production. And there is a really shift in these gray boxes. All these enzymes were upregulated, pushing this TCA cycle towards pyruvate production, really just changing the metabolism of Haemophilus for essentially to deal with this idea of nutrient starvation. So what this really taught us is that Haemophilus wasn't just dying when the nutrients were taken away. They were changing how they metabolically behave. And then some of that was at the genotype level. So we noticed mutations. I'll get to that in a second. In a story I can't tell you today due to time, but we also noticed an upregulation of all the tryptophan biosynthesis genes. And here in the bottom being the terminal tryptophan biosynthesis genes, we saw upregulation of all of these, which really suggested that there's a push, Haemophilus persistence, there's a push or a drive towards synthesis of tryptophan. And that also includes acquisition of tryptophan because Haemophilus has two importers of tryptophan itself. That's an interesting story for another time, but it's also another avenue we're following up and trying to think about ways to target that pathway as well to cripple Haemophilus. So the last part, just want to touch upon, I'll probably just highlight a few slides here in interest of time, but was this idea of bacterial microevolution that occurs during infection. So in fact, this was true, and Haemophilus was changing its phenotypic behaviors. Could we link that to any genetic changes that occurred? So we designed an experiment where we passage Haemophilus through a series of chinchillas, and these were sequential episodes of otitis media. They were 1 week long. At that point, the effusions were removed, the bacteria were isolated, then put in a second naive chinchilla, and repeated that 4 times. And what we observed phenotypically was change in pretty much everything we looked at. There was an increased bacterial burden in those bacteria that we've passaged the Haemophilus through. There was increased inflammation and fibrosis. We saw an increase in biofilm formation. There's a comparison between episode 1 and episode 4, so a clear change in the amount of biofilm. And also an increase in these intracellular bacterial communities themselves. So really striking differences that were observed as we passage Haemophilus through that chinchilla. At the genetic level, we did whole genome sequencing of the bacteria as they came out at each episode, so episode 1, 2, 3, and 4. And we noticed primarily those mutations occurred in the hemoglobin haptoglobin uptake system and also the lipooligosaccharide biosynthesis system. For interest of time, I can't tell you about these LOS changes. They're very interesting. There's a suggestion of a truncation of LOS over a period of time, which is interesting to us. However, we did see mutations in the hemoglobin haptoglobin uptake system. And I'm just going to summarize some things quickly here, a lot of work quickly. But as I had mentioned on the cartoon earlier, Haemophilus does have 3 of these hemoglobin haptoglobin binding proteins. They are phase variable, which means that's a sense of of how Haemophilus will respond to certain environmental cues, and then ultimately be able to isolates will then express a certain one of these proteins themselves. We did notice some mutations that occurred, single nucleotide mutations in the HGPC molecule, and those we've mapped now on the protein itself. You can see here in this table that the mutations were not found in just one animal of the cohort, but multiple animals. So this really told us that these were important mutations, but also we could see them in this case 5 of 6 chinchillas, 4 of 6 chinchillas, and then we could map them to episodes either 1, 2, 3, or 4 of when they occurred in that protein itself. So some of our current work then is really building these constructs through mutational analysis. My senior research scientist has generated a deletion of the two proteins that are turned off during otitis media. We know that through our work, and focus on HGPC. He's essentially built the construct with all the single nucleotide polymorphisms in there. And then we did an infection model system. And for interest of time, let me just take you right to the data. What we've shown here is that if we compete um, the non-adapted strain, so this would be HGPC that's turned on, and then we did not incorporate any of the mutations that occurred in the animal. So that's non-adapted strain, and we competed that against parent over a period of day 4, 7, and I believe 14. There was no difference. However, if we competed the parent against the adapted strain, what you can see is in all cases, there's day 4, day 7, and day 10, the adapted strain outcompetes the wild type strain. In fact, we can't even detect the wild type strain here. So in this case, those mutations that occur over a period of time conferred a survival advantage upon Haemophilus in that environment. So this just leads to our next point and our next condition. What is the clinical relevance of this? Because we know that a chinchilla is not a child. Some work had been done by other groups showing that these mutations do in fact exist in bacteria isolated from the lungs in chronic obstructive pulmonary disease, really then alluding to that point I made at the beginning of this talk of looking at different sites of how Haemophilus is adapting to different multiple environments. We've done some work now with some colleagues of ours. So with Mike Pekicaro, the Rochester Regional Health System, he has a collection of 1,300 clinical isolates from acute otitis media, really the global leader in collection of isolates for acute otitis media. At the University of Buffalo with Tim Murphy, he has a collection of COPD isolates that we're analyzing. And then with the Menzies School of Health, we haven't started work on this yet, but we're really excited to do this, really looking at strains from Australian study, looking at chronic middle ear disease in some of these patient populations as well. And it's very encouraging. I don't have time to go through all of this, but we see the point here being is that we see some of the same mutations that have occurred in our experimental model are occurring clinically in these clinical isolates. So we now have confidence that our model is predictive of some changes that we believe are happening clinically, not only in the ear itself, but down in the lungs and COPD. All right, so with that, just quick sum up. I told you 3 different stories here, really pushing towards this idea of development of competitive inhibitors, inhibition of bacterial uptake mechanisms, or looking at certain proteins and see if we can block binding and transport through these proteins, all with this idea to decrease bacterial persistence in those environment systems. So with that, a lot of people to thank, some funding that we had to do this work, and then of course to Reggie and the COMMeND program for this wonderful opportunity to share our work with trainees and the otitis media community. So thank you.
Thanks, Kevin. Very impressive. Does anyone we have space for one brief question, and then you can put the others in the chat. We can do that while Sharon, the next speaker, is putting up her talk.
Excellent talk. I have a question. Is there any relevance of anaerobic growth versus aerobic growth in the context of these phenotypic changes?
You know, that's interesting. We haven't done experimentally those types of systems. However, we do know that during infectious process that there could be a lot of changes in oxidative stress, particularly in the middle ear once the eustachian tube is closed or occluded. So that's probably some relevance to those studies, not only from anaerobic to aerobic and probably effects on metabolic activity of the bacteria, but then also I know oxidative stress starts to get into some other things with neutrophils, et cetera, and we'll probably hear some talks about that. But those impacts on driving some of these phenotypic and genotypic changes that we observe. So it's a very good question. We just don't have the model system set up to really address that at the point right now.
Thank you.
Thank you.
Okay, so if you have additional questions, please put them in the chat or reserve them for the discussion after all three speakers are done. Our next speaker is Dr. Sharon Clark, who is a Pass and William Jr. Research Fellow specializing in the microbiology and immunology of chronic bacterial respiratory diseases with a focus on otitis media. She completed her PhD in 2024, during which she demonstrated that children with chronic and recurrent otitis media exhibit reduced immune responses to biofilm proteins of NTHi. Her research aims to unravel the immune networks that contribute to poor host responses and biofilm persistence in otitis media, with the ultimate goal of informing development of novel therapeutics and preventive strategies. Take it away, Sharon.
Thanks, Reggie. So I am an immunologist, not a microbiologist. So I'm interested in how our body responds to NTHi. And so, as you said, I'm an early career researcher in the bacterial respiratory infectious disease group as part of the Wesfarmers Centre of Vaccine and Infectious Diseases at the Kids Research Institute Australia. And I'll be talking about understanding NTHi immunity in otitis-prone children. So, there we go. So I will touch on NTHi presence in otitis media, including our team's most recent study. And I'll also give some background around NTHi biofilms in the middle ear, because this is where I focused my immune studies on. Then I'll jump into the immunology and look at antibody responses to NTHi biofilm antigens, followed by the B cell responses, and then the systemic and NTHi specific cellular immune responses, which will probably be a little bit familiar to those that were at ISOM recently. Before I start, I would just like to do an acknowledgement of country. So this is customary in Australia because the lands in which we work and live were traditionally owned by Australian Aboriginal people. So I'd just like to acknowledge the Whadjuk people of the Noongar Nation, who are the traditional custodians of the land in which I'm meeting with you from today and where this work was conducted, and pay my respects to their elders past and present for their ongoing wisdom and guidance in our work to improve the health of all children. So NTHi presence in otitis media. So Kevin touched on this as well, that NTHi is found in approximately 50% of middle ears of children with chronic and recurrent otitis media, followed by approximately 30% for strep pneumo, and then 20% in others. And this was shown in the 2016 review, looking at bacterial presence in otitis media. Part of that review included our early surveillance study back in 2007 to 2009. So this was during the era of PCV 7 in Australia. And in this study, we were able to show that NTHi was the predominant cause of recurrent acute otitis media, aligning with that global average of 50%. There was still a non-vaccine pneumococci found that were quite common. During this study, we were also able to develop molecular tools such as PCR to identify the pathogens. And this has been key for us because PCR can often help find those bacteria that are found in biofilms, which can sometimes be missed through culture and has seen a much higher rate of positive bacterial species identification. We also had very little evidence of immune dysfunction to vaccines, but we didn't do a lot of studies into NTHi immunity. And we also found that when NTHi was in the middle ear fluids of those children undergoing ventilation tube insertion surgery, they were up to 3 times more likely to require further surgery. So in Australia, it's estimated about 25% of children that go in for vomits or VTIs will need a repeat surgery due to reoccurrence of disease. But ultimately, that study was now 15 to 20 years ago. So we have recently repeated our surveillance study, collecting samples in 2022 and 2023. We've collected a wide range of samples which will allow us to study both the bacterial presence as well as immunology and the immune response as well. Today, I'm just going to talk to you though about our measurement of bacterial presence in the middle ear effusions and nose swabs, which was part of our recent publication earlier this year. And just to touch on, so samples were collected by research assistant Joe Bayliss, Dr. Christina Gamez, and Danielle Headland. And the project was overseen by our program manager and postdoc, Dr. Elke Sepinen, who's the first author on this paper. So I won't go into too much detail with this because it is published, but we do see across the board in our cohorts that our cases often have slightly younger than our control children, and we see higher rates of daycare attendance and pacifier use and antibiotics in the last month in our children that have chronic and recurrent otitis media. Unsurprisingly as well, we also have more acute otitis media episodes, and up to 15% of our cohort actually had a previous ventilation tube insertion surgery before our study. So into the microbiology. We measured bacterial presence in the nasopharynx to compare between cases shown here in teal and controls shown in the black, with each dot representing the concentration of bacterial DNA in the sample collected for each child. And as you can see, in 100% of the noses, we did find bacterial DNA, which is not really surprising. And we saw higher densities of Haemophilus influenzae in our cases versus our controls. And we expect that the majority of this would be non-typeable Haemophilus influenzae, NTHi. And further to this, we also saw a much higher rate of positive samples in our cases as well, at 82% versus 47%. For strep pneumo, we did see a higher density in the nasopharynx compared to our controls, but there was no significant difference in the proportion of positive samples. We saw really high rates of positive samples for Moraxella catarrhalis. Again, that was quite similar between the two groups. We had low rates of detection for group A strep, Staph aureus, and Pseudomonas as well. But while nasopharynx bacterial presence is interesting, obviously we look at the ear, so we want to know what's happening there. So we only collect middle ear samples from our cases. We do not get middle ear effusions from healthy children. And again, we had a majority of the samples which were positive for bacteria, up to 99%. And we had, this has been moved, shuffled down, because it should be Haemophilus influenzae, not NTHi, because that's all our can categorize it as. And it was 53% of those samples were positive for Haemophilus influenzae. So this was the highest rate of species-specific bacteria we found, which was followed by Moraxella catarrhalis and then strep pneumo. And we saw, again, really low rates with group A strep, Staph aureus, and Pseudomonas. So this data was looking at the overall child. So when children had 2 ears, we combined them. But when we separated this by ear to see how often bacteria were found together, we found that Haemophilus influenzae was found in 28% of the ears and was only found with strep pneumo at 8% of the time, and Moraxella catarrhalis at 7% of the time. We also did find that Moraxella catarrhalis was by itself only 11% of the time, and strep pneumo at less than 2% were single positives for strep pneumo. So this reinforced that Haemophilus influenzae is still the predominant pathogen being found in Australian children suffering from recurrent and chronic otitis media, and that Therefore, finding improved methods for prevention and treatment of chronic NTHi infections could help reduce the impact of otitis media. So supports the ongoing work we have. I just also wanted to quickly touch on this, that in Australia, we see Australian Aboriginal children, who are our First Nations children, often have much higher rates of otitis media compared to non-Aboriginal children. And we had both Aboriginal and non-Aboriginal children in this study. This bit is not actually published yet. But we do see, even though they have much higher rates, that they've got similar levels of detection for our three main otopathogens in both the nasopharynx and the middle ear effusions. So NTHi biofilms in the middle ear. So Kevin also touched on biofilms briefly, but for anyone unfamiliar, biofilms are a structured community of bacteria embedded in a self-producing matrix, which also includes host components. They exhibit increased resistance to antibiotics and the host immune response compared to planktonic bacteria, although bacteria freshly released from a biofilm can actually be more susceptible. And there's multiple layers of defense for the bacteria that will be in the biofilms, which include altered phenotypes, the lower metabolic rates, as I said, increased antibiotic resistance, measured up to 1,000 times, and they can be inaccessible to the antibodies and phagocytes, which is where that increased resistance to the host immune response can come in as well. And so the process is basically the bacteria will attach, they end up growing up into these like clumps, and then they can become detached and cause reinfections and seed biofilms further down the track. And so this is why it's considered a major mechanism for chronic and recurrent infections. So our team was one of the teams that was able to show biofilms within the middle ear, although we are not the only team. And this was done by Dr. Ruth Thornton. So she used backlight staining of middle ear samples, where she was able to show that even in culture negative effusion, most of the bacteria that she was finding were still alive. So this falls into that why we think PCR is a better method for detection over using the traditional culture methods, because the bacteria in these biofilms sometimes don't seem to be turning up when we culture. As you can see in the figure B, there were live bacteria and microcolonies which were associated with DNA stranding. She also used fluorescent in situ hybridization, otherwise known as FISH, where we could stain using probes which were specific for either universal bacteria or then at a species-specific level. So she was able to show that 95% of the middle ear fluids were positive for bacteria. This was at a time when we were still using culture, so this rate was a lot lower, and that 65% of the samples were positive for Haemophilus influenza, presumably NTHi, being found in these biofilms like I have shown here in figure C. Although what figure C doesn't show is that many of the bacterial clusters were also frequently polymicrobial, so either including other otopathogens or just positive for universal probe not being tested, not turning up as a species-specific one that we were testing. In the samples, 81% of the samples were positive for biofilms and 50% had intracellular bacteria, which Kevin also talked about earlier, like we see in figure D. So these are both well-known persistence mechanisms, and it's clear that these have a major role. These are definitely present in the middle ears of children with chronic and recurrent otitis media and are a promising target for future treatments. So one of these has been by using vaccines which could target the proteins responsible for NTHi forming biofilms. So the Bacillus lab found probably like 15, 20 years ago now that pill A has a major role in biofilm formation and maintenance, adhesion, infection establishment, and motility as part of the type 4 pilus. At the start of my PhD back in 2017, these were actually being included into potential NTHi vaccines. So the first was from GSK and combined multiple NTHi proteins including, along with RSPLA, it included 2 NTHi adhesion proteins, protein E and protein D. It also included a protein from Moraxella catarrhalis, so the other major otitis media pathogen. It got as far as phase 2 trials in adults with chronic obstructive pulmonary disease, so a chronic lung disease also associated with NTHi biofilms. However, while it was shown to be safe and immunogenic, so able to produce an immune response, it had limited clinical impact and it hasn't gone further at this stage. There's also many differences between COPD and otitis media. The second was COMMeND. So this was a chimeric immunogen which combined epitopes from protein 5 and PIL-A. This was used in the chinchilla model of otitis media, where it was shown to prevent the establishment of biofilms and break apart also those biofilms that were already established in the model. So again, it was another promising vaccine antigen, so that's why we wanted to measure naturally occurring immunity to these biofilm and adhesion antigens, especially as previous research had indicated that there could be some form of immune dysfunction, but it mostly focused on either strep pneumo or NTHi antigens involved in systemic protection like protein 4, protein 6, and OMP26. And so a lot of this work was done in the stringently defined otitis-prone children from the Pichachira lab, as well as some of the work from our lab as well. So we started by measuring antibody responses to those NTHi biofilm antigens. Oops, gone too fast. And so we did this. This work was published back in 2022, I think now. And basically, this was our cohort. We had 3 groups. We had a non-otitis prone group who had no history of chronic or recurrent otitis media or other related respiratory diseases. We also had 2 groups of otitis prone children, so we're meeting clinical definitions for either otitis media with effusion, recurrent acute otitis media, or chronic subacute otitis media. And then we're separated based on parental report as being Aboriginal or non-Aboriginal. Again, like we saw with our other cohort, we do have controls which are a little bit older than our otitis-prone children, and this is something we adjust for when we do our statistics. Otherwise, the cohorts were pretty well matched for the key demographic data. And I'm sure there's probably quite a few of you that have already seen this, but we do see that otitis-prone children, particularly the Aboriginal children shown here in the teal, have reduced antibody titers to NTHi biofilm antigens COMMeND, RSPLA, and protein D when compared to non-otitis-prone children. In the case of protein D, we also see the stepwise reduction with non-Aboriginal otitis-prone children sitting between the 2 outside groups. This, though, was not across the board. We did see here I've shown for OMP26, so one of those systemic antigens. We had very similar titers across our 3 groups, indicating there were antigen-specific differences in antibody titers, which seemed to be related to adhesion and biofilm antigens. And we wanted to investigate this further by looking if just because the antibody titers are reduced, do otitis-prone children's antibodies also of low avidity. So this would be an indication that they're not, even though they're producing low levels and low quality antibody. What we found is that this was not the case. We found otitis-prone children, again, using those same colours, had very similar levels across the 3 groups of relative avidity index, indicating that all children were still able to produce a high-quality antibody for those biofilm and adhesion antigens. What is interesting, though, is for OMP26, where we have that systemic antigen, we do see this increase in antibody avidity in our otitis-prone children. We suspect this is because these children are more likely to have been exposed to more NTHi and are likely developing higher quality antibodies on those exposures, which is what's supposed to happen. but we do not have microbiological data, unfortunately, with this cohort to test that hypothesis. So otitis brain children are able to produce a high quality antibody just at lower levels. And so we asked the question, what could be causing this? And we started by looking at the B cell response. So the B cells are the cells that are responsible for producing the antibodies. And to do this, we selected a subset of children. We had 15, we selected 15 children from each of our 3 groups, but due to sample size, we did combine our otitis prone groups so that we had just a single group for comparison. Selection was based on having enough cells available from sample collection to be able to run the experiments we had planned, and matching the demographic data as best we could. We again did end up with children that were a little bit older in our non-otitis prone children, and this is just something we need to be aware of when we're analysing our data. So here, and for the remaining data, in blue will be the otitis prone children, and in grey are our non-otitis prone children. So here is just showing the antibody titers, which I'd shown you earlier, using only those from the subsets. And as you can see, due to the way we selected them, we do see this equalising of the antibody titers, which is nice because that means we don't need to account for this when we are analysing our cellular data. But looking at the antigen-specific memory B cells, we do see that otitis prone children had significantly reduced proportions of antigen-specific memory B cells for our 3 biofilm antigens compared to non-otitis prone children, but that these were similar for our systemic antigen. Again, reinforcing that we're seeing antigen-specific reductions in the B cell and antibody response. And so we asked if this could be due to an imbalance in the global immune cell frequencies. So to do this, we designed some flow panels. I will not go through this in the interest of time and not to bore all the non-immunologists in the field. But this does show our gating strategy that we use going forward. And starting with our global levels of B cells, we pretty much see that the levels are very similar across our 2 groups, both in the overall B cells and the subsets of naive and switched memory. Thank you, Stephen. I also very much love my flow, but it's not for everyone, I've learnt. We do see, though, with our B cells that the percentage did show up as statistically significant for differences in the median, but considering that these range exactly where our non-otitis prone children range, I don't think this will have a biological impact. We then looked more broadly at the systemic and NTHi specific cellular immune responses to see if this could help understand what's happening further back in the immunological pathway. So our T cells, very much similar to with the B cells, we do see it showing up as statistically different, but these proportions are very similar between the 2 groups. In our CD4 T cells, we don't see any major differences. So CD4 positive T cells are otherwise known as T helper cells because they provide support to our B cells to produce a effective immune response. So at both the global and at the naive and memory subsets, we see that these were quite similar. Where it got interesting, though, were in our more rare subsets that provide more direct support to our B cells. So these were T regulatory cells, as the name suggests. They're about controlling and regulating our immune response, and we see that these have much higher proportions in our otitis prone children. We also saw these in our T follicular helper cells, which are the circulating version of the cells which are in the lymph node providing supports to the B cells. And we think this increase could be impacting B cell maturation and antibody production in our otitis prone children. However, what we needed to look at was antigen-specific T cells to know for sure. We were not able to do that as part of this study, but as a surrogate, we have measured cytokine responses from stimulated cells. And using the 96 hours time point, we use this as a surrogate for the T cell response. And what we saw is that otitis-prone children had increased IL-5 production, which is associated with a Th2 response, what you think of as your allergy response, whereas IL-17 response was reduced in otitis-prone children, being the small green colour. Again, leaning towards this, which shows a decrease in a Th17 response. This is what's more associated with providing that antibody response and support. So this could indicate an imbalance of your Th2 and Th17, and this would also be impacting B cell maturation and antibody production. Of course, we did further studies to specifically link these cytokines to the T cells to know for sure. Yeah, cool. The other type of T cells are cytotoxic T cells. These are CD8 positive cells, and they're the ones that are responsible for killing infected cells. So it could be important in the case of intracellular bacteria. And we see these are increased in otitis-prone children, and this seems to be mostly driven by our memory, central and effective memory cell types, and not the naive ones, which were actually reduced as a proportion of the CD8 T cells in otitis-prone children. And to be fair, this can have two types of explanations. The first is that otitis-prone children experience persistent and recurrent infections, and when we get an infection, we have more circulating immune cells, and that's what we could be seeing. The other, which is probably the hypothesis I think is happening, is that there's an altered immune trajectory, which is moving away from the B cells and the antibody response and towards this cytotoxic type response. And this could be why we're not seeing as good as protection. This is further reinforced, I think, because we see this with the NK cells as well, which are sort of the innate cytotoxic cells. We see increased proportions in otitis-prone children, again being driven by the CD56 low, which sort of represent the activated NK cells that are much higher in otitis-prone children. And we also saw this with other innate immune cell types we looked at. So we looked at DCs, which are your traditional antigen-presenting cells, providing the link between the innate and adaptive response. So these were increased for both P and M DCs in otitis-prone children. And we also saw an increase in non-classical monocytes. And this is the reason why I think that these are a reflection of an altered immune trajectory. We also looked at cytokine responses at 24 hours as a surrogate for the innate immune response. And we saw that despite the increased innate cell frequencies, we had decreased titers for IL-6, IL-8, and IL-10 cytokine reduction in response to NTHi and protein D. And we think that this could be a reflection of the cells not being able to respond as well. So, as I stated, despite increased cell frequencies of these innate immune cells, the cells that are supposed to be producing these cytokines, they're actually producing less cytokines at 24 hours after stimulation with NTHi and protein D, indicating they may be either less responsive or not functioning as they should be. So in conclusion, I've been able to show that from the blood of otitis-prone children that they have decreased antigen-specific antibody titers and memory B cells with increased cytotoxic cell frequencies and increased innate cell frequencies. And despite this, we're actually seeing decreased antigen-specific innate cytokine responses and an imbalance of antigen-specific T helper cell cytokine responses, although overall cell levels are very similar. I have recently got my RNA sequencing data back, so I'm starting to process that now to see if we can help provide the links between these differences that we've seen. But overall, I think it's already suggesting that otitis-prone children do exhibit some form of systemic immune dysregulation, particularly in response to NTHi biform antigens, and that these findings support the development of a targeted vaccine and immunotherapy. And I'd just like to note as well with our cohort that we are actually measuring these in children up to 10 years old. So we're not just talking about an immature immune response. We're talking about something that's happening long term for a lot of these kids. And I'd just like to finish by thanking everyone, including my team at the kids. I'll change that. We've had a recent name change, sorry, I haven't updated it. It's at the Kids Research Institute. And of course, all the wonderful families and their all the participants and their families without who we don't have any study. Thank you. And thank you to COMMeND for letting me present.
Okay, Stephen has a question. We'll start with Stephen Shaw.
Thanks. It was a great talk. I have two questions. One,
Yeah.
when you were looking at the memory B cells, were you able to differentiate between circulating memory B cells and tissue resident memory B cells? And was there a difference between those two groups at all?
Yeah, so everything we've done is all circulating, so it's come from the blood. We haven't done tissue resident at this stage.
Gotcha.
We might be able to do that with some of our newer studies that have samples that will facilitate that.
And then secondly, because we get samples from our own lab gets samples from when children are having the surgeries where we do collect blood in our lab, and then we also get cells from the middle ear. I would be interested in knowing your flow panel.
Yeah.
Especially because we have been looking much more at innate cells, but as a fellow in our medical fellow in our lab is going Taylor in a more reoccurrent infection in the mouse model, we're going to try to complement that in our human data to get into more adaptive cells. So currently we kind of just dump all the adaptive cells out, which you would probably be horrified to learn.
Yeah. Yeah. That's terrifying. No, it's all good. We all have our own cell types, and to be fair, I focus more on adaptive than innate myself. But yeah, my email is at the top of that slide. If you want to reach out, I'm definitely happy to be sharing resources and that around our neurological studies and sharing the flow panel. It's hopefully going to be published at the end of this year, but there's a few papers that are sitting on my list to do that with. So see how we go.
Thank you. It was a great talk.
Yeah. Okay.
And Steve Pelton has a short question. Are the OM prone and OM free groups matched for breastfeeding?
Yes, they are in this cohort, and we see very high rates of breastfeeding in both groups. I think in the one we've just released, I think that might have had I know that also didn't have a difference in breastfeeding, I don't think either. Yeah.
Is that okay, Steve? Does that answer your question?
Yes, that's fine. Breastfeeding obviously has an impact on immune maturation, and that's why I was asking to make sure that they're matched for breastfeeding.
Yeah, so they are matched for breastfeeding, and yeah, I suppose it's also, but we don't know the long term is like, because we are looking at older children. So the youngest child we look at is 2 in our cohort. So they're not current breastfed children either.
All right, I'll introduce Ken while he's putting up his slides. And you can reserve some more questions, either put them in the chat or for the discussion later. So, Dr. Ken Brockman is an assistant professor in the Department of Microbiology and Immunology at the Medical College of Wisconsin. He's also our current ISOM secretary. His research focuses on understanding how NTHi adapts to the host environment and evades immune responses, and his lab uses models of infection and bacterial genetics to dissect mechanisms of immune evasion and persistence, with particular focus on phase variation and epigenetic regulation. In this talk, Dr. Brockman will discuss recent work uncovering how NTHi modifies its gene expression to persist within the host, and how these insights may guide the development of new vaccines targeting this versatile pathogen. Thanks, Ken.
Thanks, Reggie. Yeah, so Kevin and Sharon gave a really nice introduction to this. Hopefully I can tie some of it together, really highlighting the bacteria put a lot of effort in trying to survive and persist within the host. And the host is trying to put a lot of effort into responding to that to mitigate disease. And there's obviously this kind of back and forth pull that causes the manifestation, that manifests the disease itself. So I won't belabor this. I know you guys had a nice, session a few sessions ago about vaccine development. But just as a reminder, you need to understand the molecular basis of the disease from both the host and pathogen side. You need a good model system test efficacy, and several of those have been talked about today. And then you also need to identify vaccine candidates that are produced by, all of the bacteria that you're targeting, that also induce formation of antibodies that will be protective. And, this is really the main key point that we're talking about today. One of the big struggles with a vaccine against non-typeable Haemophilus influenza, as opposed to, the serotype B that's been pretty successful, is that despite having a relatively small genome and number of genes, and really the core genes are even smaller in Haemophilus influenza, there's a lot of adaptability and versatility provided through a variety of different mechanisms of regulation that make this bacteria really difficult to target, all of the strains, at the same time in different populations. So there's a variety of different mechanisms that NTHi can use for regulation. The first being kind of classical regulation or direct regulation. Bacteria sent some sort of signal, nutrient, other factor that's picked up inside of the bacterial cell that causes the transcriptional factor to change gene expression. You get a protein turned on or off in response. A second form is antigenic variation. It's out of my way. Which many bacteria do. So it's genetic alterations that can change the amino acid sequence of typically surface-exposed proteins, that changes, the epitope. It can make antibodies that are specific for one particular protein. Those antibodies no longer work when there's minor changes in that amino acid sequence. However, the proteins often work functionally similar. Kind of the classical example in NTHi would be the HMW or high molecular weight adhesins. So in NTHi, they're typically encoded or classically encoded by the HMW1A, 2A genes. Antigenic variation occurs through a combination mutations in these, in a very strain-specific and very high frequency. And these proteins, while they're immunodominant and were initially sought after as good vaccine candidates, due to this extreme variability, it makes them very difficult targets for both the host to detect as well as vaccine strategies. And this work, was done a few decades ago by Steve Berenkamp, Joe St. Gem, pretty heavily. Here on the right, I'm just showing this is an example of the diversity of this single gene amongst a variety of NTHi isolates in clinics. So you can see there's a lot of diversity amongst these different strains within this single gene. And in many of these genes and proteins, it's concentrated within an outer surface exposed loop that's highly variable. And there are conserved regions of many of these proteins that are critical to their function. Those are oftentimes secluded or not easily accessible from the outside surface of the cell, making it difficult to target those in an effective vaccine. Another mechanism that Kevin alluded to briefly is phase variation, or a high frequency reversible on-off switching in gene expression. Typically, there's a few different mechanisms or methods this occurs. One of the most common is you have these regions of tetrameric repeats within the coding region of the gene. When you have a certain number of genes, certain number of these repeats, your gene is in frame. You get transcription, translation, all the way through, and you get a protein product produced. Because of the high frequency of these highly repetitive tetrameric repeats, you can have a shift, increase, or decrease in that due to slip strand mispairing, due to the inability of the DNA polymerase strand replication to properly get the same number of repeats. When you have a shift, as example here, a decrease, this shift actually changes the reading frame, introduces a premature stop codon, and you no longer have this protein produced. Because this phase variation occurs due to an issue in DNA replication during each bacterial cycle, it's not believed to generally be done in response to some sort of outside factor, but this is occurring constantly, kind of at random events. So every time the bacteria replicate, which is less than an hour, there's a chance that any of these genes are going to turn on and off at random. Um, and then what they found is generally there's outside selective factors that can select for populations that have a certain fitness advantage based on what these conditions are. There's several dozen of these proteins, the iron ones that Kevin had talked about earlier. The other kind of classic examples are the LOS modification proteins. In this particular example, LICA1 is a choline kinase that adds phosphorylcholine here as a decoration to the LOS. So it's not a critical molecule for LOS to function, but it does provide a variety of different, factors that are both beneficial to the bacteria as well as potentially non-beneficial. So Jeff Weiser, Kate Fox, a few other people, have published on this fairly well. All of these proteins shown here in red are known to be phase variable in a variety of different strains. In the case of phosphorylcholine addition, when it's in the on state, when I refer to on, that means this protein is being produced and fully functional. It promotes binding to platelet activating factor, which increases invasion. So in certain conditions, as we heard about, earlier this morning, invasion can be critical for these bacteria to survive. However, it can also lead to higher immune recognition. It's highly immunogenic to have this phosphorylcholine molecule, and it's not always beneficial, particularly when these bacteria are just trying to colonize. So those populations in which it's been turned off, they actually have reduced recognition by CRP and complement. So they're better able to survive outside of cells when they don't express this. So you can see there's this dichotomy of based on the conditions they're in, having this protein expressed can be beneficial, and other times, potentially not beneficial. There's a variety of different proteins in NTHi, as well as many of the other pathogens of bacteria that use all three of these mechanisms. Over the past 10 years or so, a lot of my work is focused on an adaptation of phase variation, or a fourth mechanism that NTHi and several other bacteria use. The basis for this is a gene called MOD-A. So it's a component of a type 3 DNA methyltransferase restriction enzyme system. This gene encodes the MOD-A protein, which is a DNA methyltransferase that does site-specific methylation to the bacterial chromosome. So here in this cartoon is a bacterial cell, just an example. You have the DNA inside of the cell, and I have two different proteins here on the cell surface. What can happen is this MOD-A protein randomly gets phased on. The protein is produced. We get site-specific DNA methylation. This methylation causes an epigenetic change in the expression of numerous genes across the chromosome. They're not physically linked or connected, and that causes the surface proteins that are expressed to change. This is reversible, so they can phase back off during replication. You lose this protein, subsequently lose the methylation, you go back to this previous expression set or pattern. And what this results in is two otherwise genetically identical, but phenotypically very distinct subpopulations of bacteria within a single strain. This has been identified in a variety of different pathogens. Hopefully you can note here that they are all exclusively human mucosal pathogens that this mechanism has been identified in, suggesting that there's some sort of advantageous, reason why this has either been maintained or has evolved over time. In part because when you think of, the life cycle of NTHi, it's a commensal in the nasopharynx. You can get to the lungs, it can cause disease. You can get to the ears, it can cause disease. It can get inside of an epithelial cell, it can get into the blood, and cause meningitis and other factors. And they have to be constantly ready to adapt to these changes. There isn't time necessarily to respond cause changes to transcription, to translation, to change the protein surface. So there's always a subpopulation of these bacteria, we believe, that are prepared to survive in a variety of different conditions. And based on where the population is at, some will survive and persist and some won't. So sort of a contingency backup plan. I won't go into depth with this, but we've looked at a variety of different disease-related phenotypes that we know within NTHi are regulated by this mechanism very broadly. One being biofilm formation within a variety of different conditions. Of note, particularly the conditions of the middle ear, this mechanism seems to be most active, which is 37 degrees and a pH of 9. That's commonly found in the more chronically diseased ear. Adherence is affected differentially to different cell types, as well as resistance to a variety of different antibiotics, reactive oxygen species, both exogenous and within innate immune cells, and opsonization can obviously also be infected. So if you have a particular target on the cell surface, if that target goes away, you're no longer going to get proper opsonization and effective killing by the host response. So one of the questions, when I really started out in my postdoc working with Lauren Bakelitz, is, that's great. We see all these in vitro, but how about NTHi-induced otitis media? And I think most of you guys are familiar, but just briefly, we use a chinchilla model of otitis media that Kevin introduced earlier. There's several benefits to this. We can use a lot of the same tools that are used clinically with a child. You can see the eardrum, directly to assess disease over time. You can perform tympanometry to look for at least the movement of the eardrum to get some of the same signals that a pediatrician will use to assess, clinical otitis media as well. We also have a unique factor because of the position of the bulla here, or the middle ear, reaching the top surface of the skull. We're able to aseptically get a needle in there, and we can deliver sample or bacterial infection, as well as collect samples without having to go through the eardrum specifically. So we can do serial collections over time and serial challenge and introduce different bacteria over time using this model. So the first study that we looked at, just to try and assess this, the chinchillas were challenged with NTHi, a variety of different strains. We use a pretty low dose, so we're talking approximately 750 bacteria go into the ear. Part of the reason for that is normally in the case of an intact tympanic membrane, the bacteria ascend the eustachian tube, and they come in relatively low numbers, and then they replicate and cause, the infectious process. If we were to dump a large bolus in it, it wouldn't necessarily mimic as well what we believe happens traditionally in a child that's getting normal ascension. We use 3 different strains of our 3 different, variants, populations of NTHi. So the one that I'm referred to is 723 MADI2 on. Fast switching was greater than 90% on population. So of the 750, greater than 90% of those were on at the time of challenge. We have an off fast switching that was greater than 90% off at the time of challenge. And then we also have one that's much slower at the switching process. So it's predominantly off at time of challenge, and it takes much longer, at least in vitro, to switch. We're able to collect serial samples from the same animal over time, up to 14 days, to look at the on-off ratio to see if there's any sort of selection within the middle ear. And then at day 22, which was the end of our study, the animals were euthanized, and we could look inside and assess signs of biomass and perform histology. So our first question here was, we see in vitro that there's selection or adventation to the on or off status based on the conditions that are presented. Here I'm showing you each one of these little dashes is the ear of one of our chinchillas that we looked at. If it's blue, it's greater than 60% on. If it is this orange color, it's greater than 60% off within the animal. We found that those that we put in on stayed generally on within the middle ear and or cleared, which you can see here with the black. We weren't able to get enough sample from these animals. Those that went off, they were able to switch readily. We found a clear selection within really 5 to 7 days for the on status. which suggested to us that at least within the middle ear of the chinchilla, on provides some sort of fitness advantage to these animals. And then our slow switching, at least within the course of the 14 days that we looked at, wasn't able to switch on. So we're able to look at all three of these conditions. Here I'm showing an example of a healthy middle ear. So once the animal has been euthanized, we can dissect the ear out. Off the back of your screen would be the external auditory ear canal. Here's your tympanic membrane, and this would be the middle ear bulla space. In a healthy animal, healthy child, this is normally air-filled. You have a very thin layer of mucosa here. And in the chinchilla, there's these bonus septa here that I'm going to point your attention to that you can actually, we use to gauge the depth somewhat, where you can see this is somewhat of a hollow pocket-filled space. When we looked at 22 days, comparing these three different animals, you can see the on that stayed on had a relatively mild disease, which is, I would say, more typical or classic for an NTHi only acute otitis media infection. Similarly, those that went off and stayed off have a relatively mild, minimal amount of biomass within these ears. But what's really striking to us is those that went in off and shifted on, we see this large amount of persistent inflammation that goes well beyond what you would expect for an acute otitis media, because we're at day 22 here. We can quantify these by blind scoring, and we see that overall, those that were able to shift from off to on had significantly more biomass, as well as other signs of clinical disease. So we hypothesize that there's something about the host immune system that's uniquely recognizing and responding to each of these subpopulations, where if they go in off, they flip on within the middle ear, despite being a single strain that we put in. We thought that maybe the immune system is seeing this as a secondary infection. So it's almost like one infection, you get a new infection while you're still dealing with the first one, and the immune system doesn't know how to respond to it, and you get this over-inflammatory process. So to tease this out, we then wanted to look at the individual populations. We performed a very similar study, but in this case, we're actually able to genetically modify these so they're not able to switch. We get rid of the repeats, and it's either MOD is always expressed, which would be the locked on, or it's not able to express MOD at all and locked off. We don't get this switching. Performed a similar study to what I talked about before. Unfortunately for Stephen and Sharon, this is the only flow plot you're going to see, but I'll be showing the results of flow data. So at day 14, we collected the cells from this middle ear, and we looked at both the bacterial response as well as the host response to see if we could identify any differences based on a peer population. We first look at the macrophage response. So neutrophils and macrophages are your predominant innate immune cells, within otitis media in the first 14 days or so. I will summarize some of this data from our early work, but the first part we looked at was we were able to physically sort macrophage from within the chinchilla middle ear at day 14. We performed QRT on these samples. What we found is that pure macrophage isolated from the middle ears of animals that were infected with the off population had significantly greater relative pro-inflammatory cytokine expression compared to those that were affected with on. And this is normalized to pure cell populations. This is not bulk cross. This is the transcript per macrophage-like cell. We then wanted to look at phagocytic responses in vitro. So we've done this with human primary MDMs as well as chinchilla primary MDMs. We add the bacteria to the macrophage, let them go for 30 minutes. In some cases, we had gentamicin to remove or kill extracellular bacteria. Some cases we don't. And then we're able to measure both cytokines as well as bacterial counts to really tease out this interaction between these two subpopulations. We did, when we looked at pure cytokines, previously I showed you the transcript levels. Here we're actually looking at, after 16 hours in the culture dish, the amount of soluble cytokines within that dish. And while it's not statistically significant for all of this, we do again see that there seems to be a greater production of pro-inflammatory cytokines when they're exposed to the off population versus the on population. We also looked at killing. So this is survival after 16 hours. We put in the similar number of NTHi in a dish with these macrophage MOI of 10. And then after the 16 hours, we counted the remaining total viable bacteria within the dish, extracellular or intracellular. And we see that the off that induced more of a pro-inflammatory response were killed significantly better than the on, suggesting that either the off are too pro-inflammatory or the on are doing something to reduce the immune response of macrophage and thus would be able to survive better in the presence of macrophage. So our model for this would be that in the presence of off, we get activation of macrophage, recruitment of additional macrophage, and essentially effective killing. Or in the case of the on, you get minimal or reduced response by these macrophage. So as I mentioned, the other main innate immune cell player here are neutrophils. So we hypothesized that we'd probably see a similar response with neutrophils as well. We did a similar experiment. We isolated from those same animals the neutrophils by sorting. We performed QRT to look at a variety of different markers. The two main pro-inflammatory markers we looked at for neutrophils were neutrophil elastase and myeloperoxidase. And hopefully you can appreciate here, in contrast, the macrophage, where the off seemed to induce more of a response, the on seemed to be inducing more of a response to the neutrophils, suggesting that the neutrophils are better able to recognize or at least respond from an activation standpoint to this on population compared to the off. And then we look at killing. We see that that's again a similar observation where the on are able to be more effectively killed by isolated neutrophils relative to the off. And again, we've done this with several animals as well as human peripheral neutrophils, and we see the same effect. So what we propose here is that unlike the macrophage, when you have just the bacterial populations and neutrophils in a dish, put the off in, we get minimal response. However, if on are presented to the neutrophils, they become activated, you get additional recruitment, and you get killing. Macrophage, we think we've worked out that most of it's phagocytic based, so there's phagocytic uptake and killing that occurs. Neutrophils have a variety or a larger, tool set to kill bacteria. So they can phagocytose and kill. You can get degranulation, so you get release of reactive oxygen species, other factors, and you can also get netosis, the release of this extracellular DNA and a variety of other compounds that's very effective at killing bacteria. But it's also one of the main factors that leads to pus. So a lot of pus is neutrophils that have died. They're undergoing netosis, and you get this very thick, dense material. So we hypothesize that maybe what we're seeing here in the case of this persistent infection is a mass amount of netosis. And that combination of netosis and bacterial biofilm formation is what we're seeing this mucosal biomass is. And it's serving both as a reservoir for these bacteria to survive, and it makes it much more difficult for both immune cells as well as antibiotics and other treatments to get into this to kill the bacteria. And it can be a reservoir for the persistent bacterial infection. We took a chunk of this from multiple different ears out. We froze them for histology. This is just a single slice through a little bit of that. This would be right here, really the biomass to liquid interface. So this would be the liquid part of the ear, and heading down this way eventually would be your mucosal surface. It's a little hard to see at this low magnification, but these cells are predominantly PMNs. We do have some areas of pink labeling here of DNA that we think could be some extracellular DNA, some life cells, some death. You can't see bacteria here. This is what we see when the middle ears are infected with the pure off population. In contrast, when they're infected with the pure on population, hopefully you can appreciate that these look very different. And we have these really large, thick strands of what we believe were DNA produced here. And you tend to have less structured cells with clear nuclei and much more of a conglomeration of dead cell mass. We wanted to look at this further, so we performed immunohistochemistry. So again, this is from the chinchilla middle ear. It's a higher magnification. You're seeing here in blue the DNA, and in green these are GFP expressing NTHi. You can see individual bacteria here. You can see some that seem to be caught on net-like DNA strands potentially, but otherwise most of these cells seem to be round and intact. In contrast, when we look at the on population, again, there's a lot of mass destruction here. The bacterial cells seem to be less clearly defined. So we still have GFP that we think is leaking out and getting caught up in this mess. But there's a lot more general destruction. And if we take this biomass from the middle ears, we actually see significantly more viable bacteria from the off population than the on population at a 1-to-1 mass ratio. So the immune system, despite this chaos of NETosis, seems to be more effective at actually killing this population and clearing it out. So we wanted to also look at this in a dish. So we took human neutrophils, placed them in a dish. We added in either the off or the on pure population for 3 hours, and then we fixed and performed confocal microscopy. So here's an example of a top-down view of our in vitro bacterial and neutrophil interactions. NTHi, again, GFP are shown in green. DNA is shown here in blue, and then neutrophil last stage was a marker of activation or later stage activation, early NETosis, is shown here in red. This is a side view of this exact same sample here, where you can see the individual cells. And hopefully you can appreciate that we have these chunks or small groups of bacteria that appear to be inside of these neutrophils in this condition. In contrast to on, we see less bacteria that seem to be inside of the cells, but we do see these long strands of DNA, which are reminiscent of kind of a classical in vitro netotic event. Probably from this cell here, you can see the decondensation, neutrophil elastase. And these chunks of probably what once were bacteria, and many of them are dead, and maybe this one's still a little bit alive. We then took these same samples and we looked at them by scanning electron microscopy. And again, we see a similar effect. So these DNA strands here with the on that are producing nets are studded with these bacterial. Here, this green is just pseudo-colored. We have no way of knowing if they're alive or dead using this methodology. Or in the case of those infected with the off, we don't see as much NETosis, degranulation, but we do think that there's more signs of death potentially here. We think maybe these neutrophils are actually dying and being killed and not able to effectively clear these bacteria out. So just to summarize what we think is happening here in the context of the ear with both of these immune cells, when you have a pure off population, you get activation of macrophage. They're able to recruit other macrophage, essentially clear, and you get a relatively controlled mild infection. Even though you don't need the neutrophils, they're not doing a lot. When you have a pure on population, you have the on, you get more of a neutrophil response, netotic response. It's still able to maintain control infection. Again, you get some acute otitis media. It doesn't become persistent. In contrast, those when they go off to on within the middle ear, you have off, you get macrophage respond, the bacteria switch, the macrophages aren't able to respond anymore. You get neutrophil response, and you get this back and forth where the immune system just can't control. And you're getting a worsening of otitis media essentially because the immune system is over-responding because it's not able to mount an effective response against one of the populations or not. And that's what we see here. And this is interesting to us from the standpoint of bacterial persistence. And when we try and think about understanding how to create effective treatments and vaccines, understanding what targets, surface proteins, epitopes are being expressed on these bacteria is very important. One of our other really big concerns is you may have an on population, an off population that if you let that infection go in a child normally or give mild antibiotics or something like that, you'll get a 14-day infection, it'll clear. If we treat with the wrong treatments, we could actually force these bacterial populations to go in the wrong direction, and we could induce this inflammation, without trying to do so. But we're really trying to understand now what some of these different products are and find ways to effectively target both of these populations or use, multiple antigens in combination, as, Sharon mentioned briefly with that, to really understand what the host and bacterial pathogen needs so we can most effectively target these individual processes. I will just briefly thank, my group and the funding and collaborators at Nationwide. And if there's time for a question or two, I will take one.
Awesome. We are now open for questions and discussion. Please feel free to speak.
Hey, that was a great talk. I do have an interesting question that goes back to a previous one of these that we had from January that showed that NTHi is also able to take the nets and convert them into biofilm. Do you see when this is occurring with I believe was the on population, which does recruit more of the neutrophil response, where they are inducing an otosis response, that there is also a stronger biofilm that creates a resistant population?
Yeah, so it's actually the opposite. So we're doing some work on this for a grant that we have right now. Part of this difference in netotic response is due to the fact that the off population produces significantly more catalase. We've looked at that. The high levels of catalase actually cause an altered netotic response. So we do see some nets with off, but they're what are believed to be viable nets. So the DNA gets released from the neutrophils, but it
Yeah.
lacks neutrophil elastase, these other molecules. The off is also better able to produce dense biofilms that integrate DNA both from itself and from the host into its biofilms. So we think off is being selected for in the biomass, both because it's more resistant to ROS, it's not activating and killing neutrophils the same way, and it is, we believe we have some preliminary data suggests that it's able to use the unique type of DNA that it's causing neutrophils to release. So that's a great question. We're hopefully putting that out soon.
I have a quick question for all three of you. If you look at the literature, seems like NTHi is the main culprit for greater middle ear effusion in chronic otitis media. So which of the mechanisms that you describe would be tied to mucin production? Or is that a different webinar?
That's all Joe's work. Um, yeah, I don't know if Sharon has any additional stuff. Yeah, so that has been looked at in the literature, at least from a host standpoint, right? So defects in the type of mucin, stuff like that can affect it. But I do think based on different strains and stuff, they can also respond differently based on how much you have, right? I mean, we've done a little bit with that, and we know that the substrates they adhere to and they can bind to affect some of the signaling as well as their ability to persist, at least early on in disease.
I think the immune system itself probably is one of the sources, definitely for that. And I suppose it's all about getting the right balance of an immune response that is protective and not damaging. Definitely, like the neutrophils and the nets, sometimes they can cause more damage than the killing for bacteria. So it's all about that balance, I think.
Yeah, I think it's interesting. We obviously talked about bacterial pathogens here, but otitis media doesn't necessarily require a infectious agent for it to cause, right? I mean, you can have the inflammation and the response due to a variety of different reasons. So I think, a lot of it is host driven, and it's what the bacteria are causing the host to do. Whether or not they want to do it, there's a response to the bacteria, and it's effective or ineffective. And I think there's a lot of factors.
Yeah. Right. Are Are there other questions from the audience before we close? This has been fantastic, so thank you to all three of you. It does increase our knowledge and scope of what can be seen in the future in terms of NTHi. There's still a lot of complexity that's not understood. It's just highlighted in your talk, so thank you so much.